From the Donald Danforth Plant Science Center, HEC-TV, St. Louis Public Radio, and the Danforth Center present Conversations, a discussion about big ideas meeting grand challenges.
The theory of natural selection, the World Wide Web, liberty and justice for all. These completely different things all have something in common. They started out as big ideas, and they ultimately changed the world. This past summer, I asked scientists here at the Danforth Center to give me their big ideas. I asked for ideas that arose from their ongoing work. I asked for ideas that could be implemented to address a big challenge in agriculture or the environment. And if I could ask for a slide to come up, because I don't have the clicker. I asked for big ideas that would impact a lot of people, thousands of people, perhaps millions of people. And I asked for ideas that were feasible, that if provided enough resources, could move the needle. Oh, and I asked for ideas only from our trainees, not from our principal investigators, not from our senior scientists, the ones you've seen on the stage here before. These are mostly graduate students and postdoctoral fellows. Ideas from 9 trainees working in 3 groups of 3 were selected. They included a mathematician from China, a molecular biologist from Spain, and a graduate of the Plant and Life Sciences trainee program operated on the campus here by St. Louis Community College. After the teams of trainees agreed to present their big ideas, I then informed them that they would be doing so in a competition format on a big stage in front of spotlights and 300 non-plant scientists from the community. That brings us to this evening, and you're in for something special. So to get the program going, I'm very proud to introduce our Master of Ceremonies, leader in the community, and a great friend of the Danforth Center, Chip Lurwick.
Thank you, Jim. Good evening and welcome, everyone. We have a really special evening. As you can tell just from Dr. Carrington's remarks, we're doing something we've never done before. And for the Conversation series, for which many of you have attended, this is the first time we've done something where we've brought together these young scientists. And in this case, the format for tonight is we've got 3 teams of scientists. These are postdoctoral scientists, graduate students, and research associates that represent 3 different labs. They'll be talking about their big ideas. These big ideas are the things that they think are appropriate to address big challenges in plant science and agriculture today. They're going to be up here tonight defending their proposal to a panel of 3 judges that'll be up here that have never heard these stories or these big ideas before, so they're hearing for the first time tonight. Now, this is it's exciting because it's not without some consequences. The winning team will be granted a $7,000 grant to take their big idea down the road and into reality. So it's a very exciting opportunity for these teams tonight to have this. So what we're going to ask is the panelists tonight are going to actually be judging these teams on 3 criteria. The clarity of their big idea, the creativity and quality of their presentation, and the interaction with the panel. Those are the 3 criteria that judges are going to use to put together their score. But we have a 4th criteria, and that is inspiration and audience connection. Now, you all are the most qualified to make that judge, so we're going to ask all of you tonight to judge on what is the most inspiring story you've heard and what is the greatest connection you've had with these teams. So in order to do that, many of you walked in and got a clicker. And I'm going to ask you now to actually take the clicker out because we want to test this technology a little bit and make sure you're familiar with what we're going to ask you to do later. So this clicker, it's hard to read, small print. There's some black keys if you're holding it, and the keys are just 1, 2, 3 across the top row. I'd like to ask all of you now to just press 1 to test this fabulous technology. Go, press. Great. Okay, it's working. This is a good start to the evening because we need audience participation. So with that behind us, let me start by introducing our judges. The panelists this evening, first and foremost, we're going to introduce Vijay H Allen. Vijay is a local leader in St. Louis entrepreneurial community. He's the leader of Global STL, helping to attract many of these high-tech companies to the St. Louis region. Next, thank you. Next, we have Natalie DiNicola. Natalie, she has 20-plus years experience in the ag tech industry and currently is the president of DiNicola Consulting. And last but not least, we've got Tim Rogers. Tim Rogers is the founder and former CEO of Rogers Townsend, one of the region's leading advertising agencies and media companies. So with that, we're going to get started right away. Without any further ado, relax, sit back, and get ready for big ideas.
This is the information age, but we're drowning in data.
Not only is information doubling every year, but it is so disconnected.
We see a need to get far more value from information we collect, especially in agriculture.
Our big idea is a platform to unify massive amounts of information for more precise agriculture. I'm Lee Dwonkyomani from Ubon, Thailand.
I'm Nate Ellis from Ferguson, Missouri.
I'm Mao Li from Taiyuan, China. We are Team Top from Chris Top Lab.
Three people are blindfolded and asked to touch 3 different parts of an unknown object. The first person feels a coarse, flexible material and believes it to be a welcome mat. The second person feels a bristly stick and concludes it's a broom. The third person reaches out and feels a heavy tube and believes it must be a fire hose. They were then asked to discuss their findings and agree on the identity of the object. However, each rigorously defended their initial observation and could not agree. They were then allowed a second examination, but this time with the additional sense of smell. Each person was from St. Louis and regularly visited the zoo. They instantly recognized the pungent smell of the River's Edge exhibit. This time, after a quick deliberation, each agreed that they completely missed the big picture. The welcome mat was a coarse, flexible ear, the broom a bristly tail, and the fire hose a trunk. Of course, it was an elephant. As a team of plant scientists and mathematicians, we are here to address the elephant in the room. But our elephant is crop yield.
If you ask a farmer what most contributes to having a good or bad year, they'll say, it depends. It could be the seed, it could be the rainfall, or it could be the temperature early in the season. I come from generations of rice farmer in Laos and Thailand, and growing up in the restaurant business in the U.S., my family knows that it truly depends on a wide range of important factors, season after season. It's not hard to see this how severe weather, some due to climate change, affects our crops and our food supply. In 2012, 80% of the U.S. farmland experienced severe drought, an area 4 times larger than Texas, resulting in losses of more than $20 billion in farm income. That's more than the GDP of a small country. The drought of 2012 hurt our farms and impacted global food prices. In some ways, we in the U.S. were prepared for that big drought because breeders have produced corn and soybean seed that is more drought tolerant than in prior generations. But did you know that today, even with all our modern technology, it still takes 7 to 10 years to develop a new variety? This includes 4 to 5 years of testing after the actual breeding and line development have been completed. Climate is changing. Are we breeding crops that would thrive in tomorrow's weather patterns? Are we breeding improved crops fast enough? And importantly, are we providing farmers with enough tools to help them make the best decision in real time for the upcoming harvest?
At the Danforth Center and other institutes, talented scientists are working every day to figure out how microbes interact with crops, how roots function to take up water and nutrients, and how climate affects crop growth at the field level. Some are characterizing bacteria, tiny bacteria in the soil. Some are analyzing plant structure above ground as well as the hidden half below. And others take a step back further and study crop health at the field level. But when we talk to each other about our data, it's often like we're speaking a different language. We collect vast amounts of information, each at our own scales, and using techniques of our own discipline. This limits the use of available scientific information that we could use to improve agriculture. And remember, there are currently 7.6 billion people. We will have an additional 2.5 billion people to feed by the middle of this century.
Let's go back to the elephant parable. But this time, what if the elephant could talk?
Then it could just say, "Hey, I'm an elephant." So simple. What if plants could talk? They might say, "Oh, it's getting hot. Give me some more water, but not too much." They might say, "How about next year give me better soil?" Wouldn't it be amazing if we could communicate with plants? I'm a mathematician who studies plants. I see math in every aspect of nature that I study. Math is a universal language. And guess what? We can use math to communicate with plants. Here's what I mean. Plants excitedly pronounce spring is here by blooming their pretty flowers. Oak trees quietly hint at fall as they gradually turn red and yellow. When the Bernese winter hits, you see the leaflet trees. As you fly over the same forest over the years, you may see the changes in green and brown patterns. Plants are eager to express themselves through their shapes, colors, and patterns at all different scales. They are already speaking to us in their own language. We see a great need to measure everything that plants are communicating to us, and to integrate all of that information in more useful ways.
Our team consists of biologists, mathematicians, and entrepreneurs, and our big idea is PlantV, an analytical software that tackles the grand challenges of integrating massive amounts of information from plants into an interpretable language, focusing on plant health, productivity, and resources. PlantV will be able to measure and integrate all the shapes, colors, and patterns from different scales of data, as small as the microbe, to the entire Earth.
Our approach is to use math to translate plants' language to ours, to use math to know what plants need, to understand how different scales are connected, to predict what will happen in the near future, and to make better decisions that allows farmers to take better actions. we want to create a tool that will help us produce food today, and that will help us breed the next generation of crops.
Plant V will receive 3 major types of data that scientists often use but treat separately. The first at the cellular level, such as the microbiome teeming beneath plants and soil. The second at the plant level, such as shape and color from the shoots and roots. And finally, at the field to global, including imaging data taken from drones and satellites. Plant V will use the language of mathematics to convert complex numbers and equations into simple and specific recommendations for both farmers and breeders.
Here are a few examples of how we are already doing this in the top lab. We all know the importance of root for taking up water and the nutrients from the soil. But how can we know which one is better for a dry environment? Similarly, the shape of sorghum heads helps determine how many seeds and how large they are. But how would you measure the shape differences between these two? We already have incredible technology, like X-ray CT, to identify at the cellular level how plants benefit from the microbes. To visualize plants both above and below ground, math can convert these complex shapes into numbers that will help us better understand how plants interact with the environment. This gives us vital information to use in breeding programs that might result in deeper root for a drier climate or more branched sorghum heads that hold more seeds. This is progress. On another scale, from satellite images, we use mathematics to study the vegetation pattern. As we focus on South Sudan, here are 3 examples of satellite image data. The rainfall is one of the reasons for the different patterns. Turning this pattern into numbers and associating them with the rainfall could provide us an early warning of a dry year. Shapes, colors, and patterns are everywhere. Math connects those information from different scales to improve communication. But how do we include breeders and farmers into this dialogue?
Plant V will start this conversation. It extracts meaningful information from all 3 data streams and makes recommendation for scientists on what to breed for particular environments, and to farmers with understanding of when and where to provide water, nutrients, and other inputs. Our strategy is to tell people about their data in ways they can understand. Like the elephant parable, it puts the trunk, the tail, and the ears of the elephant all into one body.
Our technology will be used to break down the barriers between disconnected data. Breeders will get better quality information faster to reduce the time needed to improve seeds. Farmers will use Plant V to make more informed decisions, tailoring their practices in response to changing conditions in real time. Instead of a farmer discovering a small insect outbreak and applying pesticide to the entire field, Plant V empowers farmers to apply pesticide more economically and more precisely. This benefits both farmers and consumers.
This also benefits the world. Food is a common necessity, but around the globe, our food security is being threatened by droughts and floods, by insects and diseases, and by eroding soil. Not only do we seek to provide food for all parts of the globe, but we seek to do this in a sustainable fashion. Doubling our farmland and overusing of insecticides and chemical fertilizer will be either impossible or damaging to our fragile environment. Plant V will bridge the communication gap between us and plants, providing an open line of communication for scientists, breeders, and farmers to work together to optimize crop productivity with fewer precious resources, keeping us one step ahead in feeding the world.
Team Top, y'all did a fantastic job in explaining a very big idea. Unfortunately, now you have to deal with us. And so, a question I have for you all is based on the enormous amount of creativity you had in communicating your message. Question for you is, which one do you find to be the most creative communication concept, and how did you all come up with it?
In terms of the
presentation
slides and presentation?
Yep.
We wanted to find something that the audience can connect to. And food is important because everybody needs food to survive. And so we kind of just come up with a variety of things that people are used to, drones, satellites, and try to create something that would captivate the audience and something that's important to us.
And also, I think the idea that we were trying to nail home is that the biggest problem right now as scientists is really trying to communicate at the different levels. And so we thought the parable at the beginning was a good way to get people interested in something that they're familiar with, and then that they can understand and realize, oh, the smell of a pungent smell, people can agree on that, and that can help them decide together on what the elephant is. So I think that was one useful technique.
Great.
I really liked your parable. So one of the expressions I've heard to turn that parable around is, "Don't try and eat the whole elephant." You ever heard that? So one of the thoughts I had as I was going through it is it's such an important topic, and I do think you tied it together very well. And I love the idea of that plants are talking to us. I thought all that was great. But what I did wonder is, where do you start? You can't. There's so much there. It could be applied so many different ways. Have you thought about, well, what's the way to get an early win with this? What's a way to get people to see right away that, wow, if we can do that and it makes this improvement, how do we get the ball rolling?
Well, I mean, I don't want to speak for everyone, but I think the best that we've talked about this quite a bit, and a lot of the information that we showed already was stuff we're doing, and the Chris Top laptop is right there. And just like writers, when they write a book, you write what you know. So that's the first thing that we'd probably start with is we work on microbes, we work on whole plants, roots, and even trying to connect these two things here that we discussed is not that easy. But if we can do it with that, and come up with a pipeline and a system that's successful, then we think that we can apply it to other aspects. Because it's often, as Mao's, this is her, math is her
passion.
Her passion.
Her magic.
Is genius, and we always talk to her about biology and how we can apply it. And it's all based on shapes, color, and texture.
And patterns.
And everything has these in common. And so I feel like if we can do it with what we are comfortable in the lab, we can start applying it to other parts.
So I love the imagery too. I mean, particularly, you had so much imagery in there between the plant talking and the parable at the beginning. Talk about big data isn't necessarily so new, but you guys presented it in a new kind of way that people could relate to, and that was great. My question is, what is going on within your lab that gives you the confidence that you can do this, and what's changed that makes this possible? Because there's been data out there for some time, plants have been talking to us for a long time. What's different now that you're able to do this now, you think?
Is that because I'm here?
Good answer.
Cut that off.
That's it.
Thank
You're
God
done.
for that.
I think you answered the question. I think you can answer the question. I actually want to ask you another question. You guys have a really great team with the different disciplines that you'll have. What other discipline would you like to bring into your team that you think would allow you to go from where you are to the next level?
Oh, sorry.
Yeah, go ahead.
I mean, my passion was, so like my background is molecular biology and understanding how genes, express in different parts of tissue. Not to get in detail, but the same idea of how color and shapes and patterning and where it's at within an organism is something that geneticists have been trying to figure out the best way to do. And I would like to be able to do similar ideas, just like in the vegetation patterning, where you see different vegetation change based on how much dry and if it's more wet. The same you could apply with how much gene is expressed in different tissues. And you can combine different shapes, and you can look at gene expression. And so personally, that would be one of my things that I would like to add on.
Great. Team Top, thank you so much for the excellent answers to the questions we had and for a great presentation.
Thank you. I appreciate it.
Let's hear it for Team Top.
How are we going to feed a growing, changing world with finite resources, like water?
With over 7.5 billion people in the world today, it seems like everyone has their own opinion on how to solve these big issues. As scientists, we realize that nature has already solved many of these problems.
Our big idea is to transform the way in which we produce food by capturing what already happens in nature. I'm Ryan Del Percio from Wilmington, Delaware.
I'm Margaret Frank from Portland, Oregon.
I'm Patricia Baldrick from Barcelona, Catalunya, Spain. And we are Tim Myers from the Blake Myers Lab.
Picture a perfect place where there is no doom and gloom, birds singing happy songs, and flowers bursting into bloom. Believe it or not, this is my second time reciting those lines up on stage. The first time, I was 10 years old and a member of my middle school's chorus, singing a song from a 90s kids movie, A Troll in Central Park. Once upon a time, there was a troll named Stanley. Stanley had a magical green thumb, and anything it touched turned into a flower. When the queen of the troll kingdom discovered Stanley's secret, she banished him to a place she thought nothing green could grow, New York City. Soon, Stanley transformed New York City into a lush and flower-filled paradise. Well, we as plant scientists here at the Danforth Center would love to know the secret behind Stanley's magical green thumb, because we're basically trying to do the same thing. We want to learn how to make plants grow better, often in difficult areas, because we all need food. When I was young, I didn't quite understand the connection between flowers and food, but I did learn something. I learned that flowers are important in our world, our perfect place. So what is it about flowers that give them the power to take us to a special place, create a perfect moment, or remind us of a heartfelt memory? I think it's because flowers make us smile, and those moments are worth remembering. I always make sure that my mother has beautiful flowers on Mother's Day, and I can remember my prom date pinning on my boutonniere before the big dance. And everybody knows that flowers are the perfect gift to cheer someone up. Flowers also symbolize new beginnings. Think about that warm feeling you get inside when you see these little purple crocuses bursting out of the ground, letting you know spring is near. It can be argued that flowers are the unsung heroes of many of America's most cherished traditions and pastimes. Have you ever tried to imagine what they'd be like without flowers? With Thanksgiving just around the corner, there would be no cranberries, cornbread, or pumpkin pie. There would be no popcorn at the movie theaters, and no sunflower seeds or salted peanuts for Cardinals games at Busch Stadium. You see, we need flowers, not just as the basis to our nutrition, but because they are a part of the human experience.
Flowers are also important to farmers. For farmers around the globe, both large and small, flowers are their livelihoods. You see, flowers play a very important part in the reproductive cycle of many plants, including the majority that we get our food from. Without them, no seeds, no fruit, no food. Over 10,000 years ago, at the dawn of the agricultural revolution, ancient farmers, the first plant breeders, tried many ways to sustain and increase crop yields. Today, farmers, breeders, and scientists are still searching for new and improved methods to achieve that same outcome. Our big idea to increase crop yields is to capture the benefits of a natural phenomenon called hybrid vigor. Pat will explain hybrid vigor to you in just a minute. In order to capture hybrid vigor, we will re-engineer one of America's most important crops flower, the soybean. You've probably never paid attention to soybean flowers before, but you might want to. Their product, the soybean, was responsible for $23 billion in US exports just last year. If you're a vegetarian like me, then you're likely aware of the remarkable diversity of meat substitutes that can be molded from soy. Burgers, hot dogs, even my vegetarian Thanksgiving tofurkey. Believe it or not, this burger is made from soy. However, the importance of soy to the American diet goes far beyond vegetarian options. Nearly 70% of all food and oil products in our grocery stores contain soy. Indeed, 10% of our diet comes from soy. And for those of you who think soy is only used in our food, soybean is actually used in the manufacturing of plastics, paints, and clothing. And despite its integral importance to our daily lives, we are grossly underperforming when it comes to optimizing the agricultural output, or yield, for this crop.
Hybrid vigor is the phenomenon in which offspring outperform both of their parents. In other words, a crossbred or hybrid individual shows quality superior to those of both parents, like increased height, seed, and fruit production. Hybrid vigor is utilized in most of the foods we eat today. It has been used in agriculture for many years. For example, the first hybrid vigor was first identified in corn over 100 years ago. Since the first corn hybrids were commercialized, their yield has increased from 20 to 140 bushels per acre. For rice, hybrid vigor has helped to increase the production from 3,000 to 13,000 pounds per acre. We have a lot in common with plants, especially when it comes to reproduction. With few exceptions, flowering plants, like us, require a male part, called the anther, that releases pollen, and a female part, the ovary. Plants spread their pollen in many different ways. Some spread their pollen by wind. You may be familiar with this, when during spring, clouds of pollen are responsible for millions of sneezes. I love spring, don't get me wrong, but I am allergic to pollen. Other plants use pollinators, like bees, butterflies, bats, and beetles. But soybean makes small and discrete flowers that fail to attract pollinators, and instead reproduce with themselves. Since soybean plants do not outcross or breed with other soybean plants, they lack the benefits of hybrid vigor. We would like to re-engineer the soybean flower to make it self-incompatible by mimicking the successful strategy of other flowers, like holly, pineapple, or papaya. We have identified few key alterations that can transform the way that soybeans reproduce. These simple changes will not only allow farmers to take advantage of the powerful phenomenon of hybrid vigor, they will transform our food supply. We consider that the easiest way to do this is to generate male sterile flowers, something like this. In the past, male sterility has been used frequently for hybrid generation. The most common technique is manually removing the anthers, the male part, from the flowers. But as you can imagine, this is extremely labor-intensive and time-consuming. Can you picture going flower by flower, removing these little tiny anthers? Actually, I have experience with that. In Spain, we use saffron in most of our typical dishes, like paella, fabada, or zarzuela. That's the paella my mom and I cooked last summer. Saffron is made with the reproductive part of a very pretty flower and needs to be hand-picked one by one. Believe me when I say this option is clearly not suitable for large-scale production. The second alternative is inducing male sterility with chemicals. But the use of chemicals may have some safety concerns and environmental consequences. Nowadays, we have a new, efficient, environmentally safe, and sustainable strategy through the use of biotechnology. Scientists have already identified several key genes involved in the development of the male part of the flower in other plant species. We plan then to identify these genes in soybean and engineer them to bring about male sterility.
Since we do not want to do all of the work manually, next we will need to restructure our soybean flower so that it's attractive to bee and other common pollinators. We know that bright and showy flowers are bee magnets. So how are we going to transform our little soybean flower into a bee haven? We can mimic what already happens in nature. Scientists have already identified several key genes that control flower petal development. Using a biotechnology approach, we will focus on enhancing the function of these genes in our soybean flowers. In essence, transforming our small self-pollinator into an irresistible hub for bees. We also know that bees are more likely to return to flowers that give out sweet rewards, nectar. In most outcrossing flowering species, nectar is delivered to pollinators through a structure called the nectary. We as scientists have identified the key gene that controls nectary development. We will focus on enhancing the function of this gene in our soybean, making it possible for our flowers to attract their bee pollinators and keep them coming back throughout the field season.
The late astrophysicist and great science communicator Carl Sagan once said, "Somewhere, something incredible is waiting to be known." Well, Carl was right, and that something incredible has recently been revealed to us. Today, we know how to reprogram and repurpose an ancient tool used by bacteria for hundreds of millions of years as a way to defend themselves against viruses. This literally life-changing new technology, called CRISPR, is now inarguably the most widely used gene editing tool in the scientific community. I would like you to imagine that every single living organism has a book with detailed instructions on how to grow in different environments, how to manage stress, and how to reproduce. And every one of these books, yours, mine, and soybeans, are all written in the same language, DNA. Now, thanks to the work of many, we have begun to read these books. And with CRISPR, we are able to rewrite their stories. We will use CRISPR gene editing technology to re-engineer the soybean flower. Traditionally, when introducing a beneficial trait from one plant into another, it can require several generations of crossbreeding. That means taking two parental books, breeding them together, selecting an offspring that contains the favorable trait, and then breed and select and repeat again and again until you have an offspring that contains all the traits or pages from one plant and the single beneficial trait from the other. Now with CRISPR, a form of molecular scissors, we're able to open up a plant's instruction manual and replace a single paragraph, a sentence, a word, or even a single letter, drastically reducing the time, energy, and cost compared to traditional breeding methods. This new biotechnology has not only been one of the greatest contributors to increasing our knowledge of plant behavior, it has now become the go-to tool addressing the needs of farmers worldwide.
Our big idea will allow us to engineer male sterility in our soybean, as well as restructure its flowers and nectar content, making it more desirable to pollinators, and ultimately take advantage of hybrid vigor. So what does this mean for states like Missouri, where soybean covers approximately 5 million acres, or 120 times the size of St. Louis, or 3,800 forest parks, or 11% of the total land in our state? We estimate that by transforming soybean from a self-pollinating flower into a bee-pollinated crop, we will increase soybean production by 20 to 30%. This translates to an increase of 1 billion bushels, or the equivalent of 60 billion pounds of soybean per year across the United States. That's 60 billion pounds more food to help support a growing population. Finally, when something like soybean is so interwoven into the fabric of our society, a change of this magnitude can have remarkable impacts for our farms, for our farming communities, and ultimately for all of us who depend upon what they produce. Thank you.
Team Meyer, that was an excellent job. Really enjoyed that presentation. One of the things I really enjoyed was it's hard for folks sometimes to appreciate how much plants and agriculture affect us. And you didn't just talk about food, you talked about all the different applications in our life and really what it means for our country's economy. So that was great. I really liked the way that you went through that. What you're talking about is pretty significant. Nature is really complex. You're talking about a very fundamental process in this plant species. Tell me what gives you a little bit more of why you have confidence that you can do this, and what do you think is going to be one of the biggest complications in being able to achieve your big idea?
So I guess as we mentioned in the presentation, in other species, a lot of these genes that control the processes that we're looking to alter, nectar, flower petal development, as well as male sterility, have been identified and really well characterized in other species, as well as some of them in soy. So we believe that in terms of focusing on the genes to target to make these transformations in the flower, that should be fairly straightforward. Designing the CRISPR experiment itself also is fairly straightforward now.
Yeah, it's a good question, though, about the complications, which are a little difficult to anticipate before it actually, until you actually do the experiments.
I guess one more sophisticated aspect of the project is attracting the pollinators to make the crosses, so that there's anytime you bring in another species to interact with the system that we're trying to influence, that could become more complex. So certain pollinators are more attracted to different specific colors or scents. And so it might take a little bit more flower elaboration to get the pollinators that we want coming to our plants.
I think one of the things, so you did a great job. I think, I've heard it said that people learn through analogy and metaphor, and you did a little bit of that. But I think one of the things to pick up on what Natalie said is, I thought you gave great examples, great statistics of everything that would happen. Is there any analogy you can give, or this would do for soybeans what something else did, even in an unrelated field? Because I think part of the whole idea of being here tonight is clearly, believe it or not, you guys know more about this than I do, and I do not say that lightly. But in trying to get people to really go, man, if we could do that, and I think you did a great job. Again, numerically, any thoughts as you were going through it that you thought, if we can make this work, this would do for soybeans what something else did? Any thoughts like that?
Well, I'm the one who brought up Carl Sagan, so obviously I'm a lover
which I thought was great.
of astronomy. So a lot of the new techniques, I think even just CRISPR gene editing, I like to compare it to what the Hubble kind of did for astronomy and just seeing our place in the universe. Mm-hmm. And it allows us to dig deeper. So like I mentioned, it's not only addressing the needs of farmers, but it's also increasing our general knowledge of plants and how they interact, and not just plants, but many other species as well. So that would be my comparison.
Well, I think you're onto something describing how you made it very personal. You know, you can imagine the bees, and I think the more of that people understand, you know, because it also gets away from the too scientific GMO, all the other things, you know, that can come up. So I really like that part of it.
So, you know, you gave me a really new insight into hybrid vigor. And so when our son comes home and talks back to us, I think that's hybrid vigor. It'll make it a little bit easier to accept it now.
We'll have to add that to our list of things.
So it's clear that you all really came together as a team, but wasn't the way it started. What did you learn about yourselves that's going to help you as you now take this into the real world to make this really big project a reality?
Well, I definitely we learn a lot, not only personally, but also how to work as a team. And we all know that it's sometimes complicated, adjusting and making everything. I've learned a lot on how to do this that I've never done before. So being like here and try to explain what we do to people and try like to get the message with them home. So.
Yeah, we were definitely a team. I guess we didn't introduce our skill sets, but we're a team of very different skill sets. So I'm a developmental biologist by training, and so learning to communicate my perspective on the problem with Pat and Ryan, who have, you guys can say what you're experts in, but they have different expertise. So communicating across expertise to come up with a strategy that we thought was reasonable, and then to find the language to explain it to, you know, non-plant scientists.
Dumb people like us. Go ahead.
No. People who are very good at other things other than plant biology. Yeah. So try to translate our ideas. So.
Yeah. For example, I like to say that I am a molecular Jedi. So I've been working with genes, their function, studying them, and even engineer them in plants for a few years. So that kind of interactions that she was saying, it's like she's more development part, and I'm more like mechanistic, and I like tiny things. So.
Thank you.
That was a great job, Team Meyer. Thank you very much.
Yeah. Thank you.
Let's hear it for Team Myers.
The Green Revolution gave us what we needed to feed the world today. But what about tomorrow?
We see farming becoming more unpredictable because of things like climate change and new emerging diseases.
And it's not just about farming, but it's also about farmers and the farming communities and all of us who depend on them.
Our big idea is a better way to give farmers information they need to reduce uncertainty on the farm.
I'm Ann Phillips from Augusta, Georgia.
I'm Dan Lin from Tustin, California.
I'm Jeff Berry from Waterloo, Illinois, and we are BART team from the Becky Bart Lab.
Good evening. It looks like we've all made it through another summer. It's fall, finally, but it's also flu season. How many of you have already gotten sick? I'm from California. I'm not used to the cold weather at this time of year. I got caught outside without a coat when a cold front rolled in a couple weeks ago and ended up feeling so sick that I thought I would have to go to urgent care to see a doctor. The kind of doctor my parents wished I had become. Now, I am a doctor, but I'm a plant doctor. I try and figure out if a plant is sick and why. But why should we care if a plant gets sick? Why should we care if corn gets fusarium root rot? Why did I become a plant doctor? My parents grew up on the island of Taiwan. After the Communist Revolution in China, 2 million refugees flooded the island, resulting in food shortages. food insecurity. At times, the only thing my parents had to eat was the starchy water left over from cooking rice. That's why I'm a plant doctor, to combat food insecurity, of which plant disease is one of the greatest drivers.
Now, I have seen this. I have seen firsthand the impact that plant disease has on crop yield, because I grew up on a farm. There were some years where we had large yields, and we were all very happy. But there were some years where we had small yields, and we struggled just to pay the taxes on the land. Crop loss is something very personal to me and my family, and this is why that's why I started researching plant disease. I'm part of a growing computational community here at Danforth Center. I'm a computer scientist, a numbers guy, and I'd like to share with you a calculation I did. In 2016, American farmers brought in 15.1 billion bushels of corn. But it's estimated that about 10% of what was planted was lost due to disease. With a little math, we find that about 1.7 billion bushels of corn were lost. 1.7 billion. That's enough corn to completely cover urban St. Louis in 30 feet of kernels. Look up there. That's only about 20 feet. That's crazy, right? In today's age, is it acceptable for us to lose that much due to disease?
Just like you and I, plants get many different types of diseases, from pathogens ranging from bacteria to fungi to viruses. But unlike us, they don't get runny noses or tuberculosis or pneumonia. What they get are rotted roots, stunted growth, and wilted leaves, all of which contribute to decreased yield that threatens the livelihoods of farmers and contributes to the hunger of nearly 1 in 8 people in developing countries.
To give you a sense of how devastating plant disease can be, here's an image of some cassava storage roots. Many of you are familiar with cassava as a main ingredient for tapioca pudding. But in Africa, cassava is the main source of calories for 500 million people. Elsewhere around the world, an additional 300 million consume cassava on a daily basis. Currently, in the sub-Saharan continent of Africa, there's a deadly viral disease called cassava brown streak spreading from the shores of Kenya and Tanzania through Uganda to the Democratic Republic of Congo. This disease causes the rotting of the storage roots and can result in yield losses of up to 70%. Worse, the symptoms of this disease are so hard to spot that farmers only find that the disease is there at harvest. For smallholder farmers who depend on this harvest for income and food, this is the equivalent of showing up to the bank and finding that 70% of your savings has suddenly been wiped out.
And how do farmers respond to these emergencies? They don't call their local doctor or 911. They call their neighbor Joe, who lost their crop the year before. They do what the rest of us would do. They Google. On the other hand, they could talk to consultant companies or seed companies. But while they may have information on the most common diseases, they won't have the most up-to-date information on what's in the farmers' fields right now. Who farmers really need are experts in disease diagnosis and management. I guess the best example that we have right now are extension scientists. These scientists are part of land-grant universities, and their job is to convey science to farmers and to translate the needs of farmers to scientists like me who can help solve their problems. But here's the issue. Do you know when this system was created? 1862, when Abraham Lincoln was president, and the system has not been updated to help the modern farmer.
There are roughly 300 extension plant pathologists, extension plant doctors in the United States, responsible for nearly 1 billion acres of farmland. To put this in another way, each extension agent is responsible for nearly 3 million acres.
Now, this is exactly what happens on Little House on the Prairie with the traveling doctors. Farmers are asking for help, but the current system is not optimized to handle the enormous workload and enormous amount of problems that occur year after year. And worse, this system is spreading out the people who can do anything about it way too thin.
This system is archaic and doesn't allow for an adequate response to emerging epidemics. There's also an added layer of complexity. Just like farmers rely on extension agents also rely on scientists to develop disease diagnostic tools, disease management strategies, and also to have an underlying understanding of why diseases happen to prevent them from happening in the first place.
Now, I have seen firsthand what this communication breakdown looks like. See, I work on a disease called cotton bacterial blight. And this is a bacterial disease that affected crops decades ago. But as recently as 2011, scientists in Mississippi and Arkansas started seeing this disease cropping up again in their fields. Since then, it has spread across the United States to most of the cotton-growing areas. And now, it's as common as the common cold, cropping up in the same areas at the same time of year every year. And the scientists who first found it, they didn't realize it was an epidemic. In fact, the only way that our lab found out about this disease was through Twitter. And the farmers, they had never seen this disease before. And their neighbor Joe and the seed companies, they couldn't tell them how to solve it. So yet again, Twitter was the best way for scientists to reach out to the public. Now, this disease took 5 years to solve. 5 years. And what farmers really could have used was information on where this disease was and what varieties it was affecting.
Farmers, extension agents, and scientists each had a piece to this puzzle. It took a serendipitous meeting of all three to put the pieces together and solve the problem. This is the genesis of our big idea, that it shouldn't take serendipity to identify and stop an emergent epidemic. Our big idea is to leverage advanced communications and enhanced social connectivity afforded to us by the internet to modernize disease identification and management in agriculture.
But if you go online right now to try to find this large collection of plant diseases and what to do if you find them, you're out of luck. We would like to build this online resource. Our goal is to connect everyone in the ag community, including all of you, in an efficient way. And we'll call this database Ag Connections. This database will close the communication gap by matching the farmers with problems with the right scientists who can diagnose, manage, or even solve those problems.
Using this app will be as easy as taking a picture with your phone. A farmer can go out into the field and take a picture of a diseased plant. This will then uploaded to the database using a simple app, which will be that farmer's window into the Ag Connections network. They can then upload information such as the variety, the irrigation techniques or the chemical treatments that they use, so that we can have information on this field. This will then automatically be uploaded to the database, so their local extension scientists or disease specialists will be able to diagnose the disease and recommend next steps.
It's also possible that a disease might not be identifiable from just a picture. What happens then? This is where the networking aspect of Ag Connections comes into play. Think online dating, but instead of prospective dates, Ag Connections will suggest contact information for local molecular labs or research groups who have the requisite diagnostic technical expertise. Farmers can then send disease samples to these parties, and once their work is done, Ag Connections can suggest potential solutions in the form of resistant plant cultivars, or potential pesticide applications, or even best agricultural practices that will limit the spread of contagion.
At the same time, our app will automatically interface with geospatial networks and weather databases. This will then automatically pinpoint on a map a potential outbreak for your disease. Then it will alert Ag Connections users in the area using the tool that scientists have been using before, Twitter.
By collecting all this information would allow any Ag Connection user to track a large number of plant diseases in a variety of crops in real time. This means the people who are out there right now fighting plant diseases now have an effective way in an efficient way to manage current outbreaks. And this database is the necessary tool to prevent small outbreaks to becoming widespread epidemics.
It will also allow scientists to study how and why disease outbreaks occur, so that we can fasten the way that we study disease, and so that we don't have a repeat of cotton bacterial blight. We'll also be able to study varieties, look at how climate change affects crops, and to discover new ways to fight disease that comes with our ever-changing environment. This will then allow us to have greater food security for each one of us and our loved ones, and financial security for the men and women who make this happen.
Lastly, Ag Connections would be fully translatable to any country on any continent. This brings us back to cassava brown streak, the devastating disease spreading across Africa. If Ag Connections were to be implemented, farmers would be able to rapidly identify the presence of disease and prevent further spread. This would be real game-changing impact.
This is Anne.
Dan.
And Jeff.
And we are the BART Lab team with Ag Connections, working to bring together farmers and scientists so that we can identify the disease outbreaks of today and prevent the unnecessary crop losses of the future.
So you guys did a great job, and clearly talking about something that has incredible implications. So you might know a little bit more about plant science than me, little. I've read a few books. I've read Botany of Desire. I think that qualifies me to comment on this. But one thing I thought was very interesting is in behavioral science, loss aversion is the most powerful motivator, more than that something good can happen. So I think you're exactly in the right area. One thing I wondered about, and I think it's the whole idea is terrific. The only thing I didn't like was Ag Connections, just because, and I wondered if you had considered other names, just because that's pretty general, and that could be, that could relate to marketing or anything else. Just curious, did you talk about anything else that might get a little bit more specifically to what this is going to solve?
Yeah, we thought about more of a management section, but what we really wanted to focus on was the connection, the bringing people together. And that was really the key part of our app that would set us apart, so that we can actually bridge the gaps that we're trying to deal with.
Mm-hmm. So, your presentation was so powerful in that connection concept. I thought you guys were building the Tinder for ag. And so the question I have for you all, with this powerful concept that you have, what do you think is going to be, who's going to be your greatest ally, and where is going to be your greatest challenge in making this happen?
Well, we definitely need someone with computer programming expertise. Those are the people that we need to make this happen. But I think, like Anne alluded to, a lot of scientists would really benefit from this, and a lot of farmers would also benefit from this. So I think the buy-in from those two groups would be immediate if we brought this out there.
Don't forget, if you give me $3,500, I'll name this thing for you.
Okay. All right.
There's
We'll
no
keep in
reason why
this Well, I think that's one of the things that struck me. There's the technical aspect of this in terms of understanding these diseases and what, but a big part of this sounds like it's about socializing it across, all these different academic institutions, out in farmers' fields, in places where farmers are really hard to reach, convincing people to actually share this information with your database. How do you think you'd go about trying to accomplish that? Would you partner with people? What would you try to do?
Yeah, the very first step would probably be getting in touch with the extension scientists, because they know the local farmers. So if you can get the extension scientists on board, they're going to disseminate that information if they believe in it. And they should. So that's the first step, is trying to get the extension scientists involved, because that will branch out.
Right, there's already a network there. So we would be there to help facilitate it. And getting it into the hands of farmers is our top priority. So giving it to the extension scientists to help send it out to all of their clients is really our main goal.
So you guys, this is new for you. The whole idea of this is you've got a great idea, the science, everything, you've got that. So as you're presenting, and you know this is your first time in front of a group like this, is there anything you think, man, I wish we could have explained that a little better? Any thoughts as you're now thinking about what you just did, and you did a great job? Anything where you go, well, I wish we could have hit that a little harder?
The right answer is nah. We got it. We nailed it.
That's an acceptable answer. Dr. Carrington will be seeing you more about that. Yeah, if you're willing to settle, we can all go home.
I think the biggest thing that I wanted to convey was the reason why we wanted to do this. So I work on cotton bacterial blight, and I see this disconnection between the extension scientists and the farmers, and not everyone is being reached. And if the scientists can't figure out what the biggest problems are, then they can't solve them.
So how did you guys come up with some of the imagery, particularly Abe Lincoln? That one was really like, how did you guys come up with that? Was it sort of a team effort? You were just bouncing it around and you came up with different ideas for your imagery of things?
Oh, yeah. So we got some mentorship, and it was a team effort.
Jim played a big role in helping us with this, but I think with our imagery, we had a tech in our lab who's really good at explaining visual data. And so, Molly.
And have you thought about, what would be the first signs of success? What would have you feel like, this thing it's going to do what we want it to do? It's not going to just be something else that's a good idea.
Well, the first step would be, get a computer, right? And then after that, like we said, we want to talk to extension scientists. The very first sign of success is people starting to upload these images, right? And then the extension scientists would respond. And then ideally, people would use this database more and more. So it's a forward feedback loop. So once we get more data, we can do more things with it. So we can get more data, and then it just goes forward and forward.
And if we get this data in time so that we can actually make a difference, so like if the next crop rotation, they can plant different varieties or have different chemical treatments, that would actually make a really big impact into the farming community.
And how will you manage sort of the geographical piece of this, right? So there's going to be, how will you be able to predict what might happen based on where you see outbreaks happening?
Well, you look at weather patterns now, they're trying to do that now. Essentially, we'll just try to do the same thing. We'll see the spreading of a disease in one particular direction. You can kind of anticipate where it's going to go just based on its trajectory. So that's kind of the idea.
Yeah, and we'll also have historical data on what type of conditions these diseases typically are successful in, and then that can be overlaid with the geographical information that we have, and that'll be useful information for farmers to know what type of varieties they need to plant to avoid these diseases.
Well, I think you guys did a great job, and I think one of the other things that you made it very personal. I mean, I believed each one of you that this was this wasn't just a job. This wasn't just come up with an assignment to make Dr. Carrington happy. I think so great job.
Thank you.
Let's hear it for Team Bart. Well, that was outstanding. I know all of you would agree with me on this, but one of the most impressive things about tonight is the fact that we can all have great optimism about the future with scientists like this here and what they're going to do for the planet in the future. So now the actual really hard part, how do you pick a winner? And that is what we're going to put to our panelists at this point. So as a reminder, the panelists will be judging on the connection with this big idea and the clarity of that, how well that came across, as well as the creativity and quality of the presentations, and then the interaction with the panel. Each of those criteria are worth 25%. So with that, I'm going to ask the panelists to go ahead and exit the auditorium and deliberate in the hallway, and we'll see you in just a little bit. Now, that fourth criteria is what's your job, and that is really thinking about inspiration and which of these teams really connected with you the most. We're going to ask you all to vote at this point for which you felt was most inspiring and connected to you. So we're back to this clicker again. Across the top row of the clicker is 1, 2, and 3, and your choices are up on the board to remind you. You'll have a minute to make your selection. And for the first team that came up, Team Top, you'd click 1. For the Myers team, the second team that we have, it's 2. And then for the Bart team we just saw, it's click 3. So with that, we'll set some timer going and take a minute to make your vote.
Is everybody done voting? I hope it worked. In just a moment, if it worked, Tam is going to come out of the back door there, so make sure you clear the door. And if it did work, she's going to be holding something and looking happy. In the meantime, I am going to give a few thanks and tell you a few things about the teams. Okay, I'm filling time here. First of all, I just need to let you know that this program is part of our ongoing conversation series, and the series works because of the tremendous support and the great volunteers from the Danforth Center Friends Committee, especially the Conversations Committee, which is chaired by Von Brokaw. Von, I'm not sure where you are. Thank you so much for all the work. Tonight's program also had great help from another group at the Danforth Center, the Danforth Leadership Council. Many of our on-stage participants are associated with that group. Devin Hill was the videographer and the producer of those fantastic videos that introduced each of the groups. We're very thankful to Devin and his team. George Vasquez at PSAV is our sound engineer in the booth. Alexa Estelle is running the clicker system. Haven't seen Tam come out yet, so it's a tentative thanks. She did? I take it all back, Alexa. Thank you for making it work. HEC-TV is recording this. It will be broadcast at several points in the future. I'm sorry I don't have those times, but they're at the regular times that they always show. Our good friends at St. Louis Public Radio underwrite the Conversation series, and we're very appreciative of that. If this all sounds like a big production, you would be correct. Tam McGuire, Amanda Karras, Bill Stutz, Andrew Whithouse, the facilities team here at the Danforth Center, everyone in the development office. It truly is a team effort, and thank them. Thank them. Please join me. Now, let me tell you just a little bit about the people and the teams that you just saw present their big ideas. But before I do, I want to announce something. Because there is only one $7,000 winner, how can you not give 3 awards? But we're going to stick to it. There is one $7,000 grant winner. But for the 2 runner-up teams, they are each, because they did such a fantastic job, we're going to award them each $2,000 grants to get started on their ideas. These are graduate students, postdoctoral associates, research associates working in 3 labs. Throughout the year, we host around 100 trainees, most of them resident trainees, but many that come in for the summer or that visit from overseas for short periods of time. Training the next generation is integral to our mission. What is training? We talk about that a lot. It includes an awful lot of things, from extensive preparation to give them the tools to be competitive scientists, whether that's as a faculty member in a university, a scientist at a place like the Danforth Center, or in a company like the one across the street, or the 45 other smaller companies that populate our innovation ecosystem within an eighth of a mile of the Danforth Center. They are given training on how to write a competitive proposal, responsible conduct in research and ethics, leadership training, and as you saw tonight, and I'm really glad they really shined tonight, how to communicate effectively to both their peers and to a public audience. They're not back yet, so I'm going to slow down. Let me say this, and I do want to slow down for this part. If you liked what you heard tonight, and you think it's important that we do our best job to train these scientists and the next generation after them, consider supporting it. Consider supporting what we do to prime the next generation to express and execute on their big ideas. You can participate in support through annual giving. Many of you do. Perhaps most of you do. If you're so inspired, you can even designate gifts that have specific purpose for education, for training. Come on in. Okay, I can speed up now. You can give to this program, and every contribution is valued, and it gets put to work and invested in people like those you saw today. You're going to receive a survey by email, and it's going to simply ask you about this program, what you liked about it. It's just a few questions. It'll only take a minute to fill out. When you receive that survey, please fill it out. We use that information to improve the conversation series. I had some extra filler stuff that I don't need to cover. Lucky for you. I'm going to turn it back over to Chip.
Okay, well, this is the big moment. Thank you, Jim. Thank you very much. Well, this is it. Obviously, there's been a lot of build-up. We've had some great material by Jim. Vijay's doing 5 minutes of stand-up after that you might want to stick around for. But at this point, let's invite all the teams to come up onto the stage. Come on up. So this is the big moment, and that moment's really going to be owned by our panelists. So I'm going to turn it back over to you, Vijay, Natalie, Tim. Why don't you tell us the outcome?
So great job, teams. You'll really hit the ball out of the park, which means our job was harder. And it really took a lot. That's why we took every minute that Jim gave us to come up with what we thought would be the best way to do justice to the great work you all did. What happened tonight was not only entertaining, but really, really important. Because if you look at the big challenges we face as a society, we have the issues of climate change, or vaccines, or GMO, or evolution versus creationism. And in the scientific world, there is no doubt. There is crystal clarity. You go into the lay audience, it's a toss-up. And if we can't resolve that conflict, we are going to be really struggling with this. And so if you look at survey after survey, who does the layperson look up to with the most credibility? It's a scientist. And yet today, scientists are really challenged with being able to communicate in a way that people on the street can understand. So what's happening today is we are training the scientists for the 21st century role of not just being great scientists, but being great communicators. And that's why I give Jim and the team at Danforth a lot of credit, and you all for being the folks who took on this very challenging role. You came here to do great science, but we put you onto another challenge, and you did a great job with that. So that's why this is both entertaining and important. Thank you.
And I just want to kind of echo that, but I want to point out how hard we all think it is to do public speaking ever. But to do this, they didn't get to talk about what's sort of in their day-to-day wheelhouse, data analysis, you know, the kind of peers they normally talk to. They had to come up with humor, and they had to share parts of themselves with us and help us understand some very complex processes, and did an outstanding job. So thank you. Congratulations to all of you for that.
Well, my role, as I think you saw, was to represent empty promises and getting up in front of people where there are no facts. I'm from the world of marketing. So, but I think to go back to what a couple of people said, and I think it's really true, that one of the things I've mentioned to several people here as I've been involved with the Danforth Center is that growing up, I was never good at the sciences. Frankly, I did the minimum I could do to get on to the next thing because I knew I was never going to be very good at it. And I think that what something like tonight does is it's for people like I think most of us, certainly like me, where you realize just how important all this is, and we let these fantastic talents tell us in a way that makes it so personal to them that I think it's unforgettable. If you would see this in a different setting or see a paper, it'd probably go right over your head. But when you can see the heart and the passion and the soul that all of them have put into this, it's just a fantastic event. And just, I can't thank you enough. It was fantastic. So I also, I get to be the voice of all of you, and the vote is in, and I want to thank you. Apparently, I'm the tallest, darkest, and best looking, so thank you very much for that. That was number one in your hearts. So, but the vote, which, as we've talked about, was a quarter of our vote, was for which team that the audience came away feeling did the best job with just kind of, the way I would put it is, easiest to explain to somebody when they leave tonight and they say, what did you hear about? Well, this would be the thing that would be easiest. And the winner of the audience vote was Team Bart. So congratulations.
And now I get to announce the actual big idea winner. As you can imagine, this was an extremely hard decision for us. I think all of us here are very proud of the Danforth Center to have such a premier research institution right here in our own community. And these young scientists, without a doubt, exemplified the quality and the passion that the center stands for. We were combining your audience vote with the criteria of the clarity of their big idea, the creativity and quality of their presentation, and how well they interacted with our panel and essentially were able to think on their feet. And like I said, it was a lot of deliberation, but I'm excited to announce that the winner of Big Ideas
Really?
2017.
You're going to tell it now. You're going to tell it.
Is Team Bart.
Yeah.
Congratulations, and let's give a big round of applause to Team Bart and all the scientists for an outstanding job. Thank you all for coming. An incredible, incredible result.
Thank you.